Well, thank you so much for the very generous introduction. My name is Joe. I'm, like you just heard, I'm a fifth-year PhD student in Andy's lab. Andy asked me to apologize on his behalf. He wishes he could be here, and he sincerely appreciates the invitation. I'd like to thank the organizers on my behalf as well. This has been a beautiful symposium. I'm learning a lot. The venue's beautiful, the city's beautiful, and it's quite an opportunity to be here. So I'll be presenting on our work, developing physical models of chromatin organization. We're taking a slightly different approach to studying chromatin in that we use fundamental principles from statistical mechanics and polymer theory to predict how chromatin will organize based on patterns of epigenetic marks. So I'll jump right in. I want to begin with some brief biological background. For context, we have about 10 billion miles of DNA in our body. This is enough DNA to take us to Pluto and back, and this equates to about 2 meters of DNA per cell, which compacts into a nucleus that's only about 10 microns in diameter. This high degree of compaction is achieved by wrapping DNA tightly. I might give up on the pointer. By wrapping DNA tightly around histone octamers to form units called nucleosomes, which then coil into tight chains that make the chromatin fiber. Despite the high compaction of our DNA, we need precise control over which genes get expressed. And this is because we have a diverse array of cell types that all share the same underlying genetic code. And a large part of controlling gene expression is controlling which genes are accessible. So we know that genes in transcriptionally accessible regions tend to be transcribed, while those in tight regions tend to be suppressed. The structure of chromatin is dictated by patterns of chemical modifications on the DNA and the histone octamers called epigenetic marks. These epigenetic marks are preferentially bound by reader proteins, which interact with one another to affect chromatin structure. During this talk, I'll focus on one prominent repressive epigenetic mark, H3K9ME3, and its associated reader protein, HP1. HP1 has two important domains that facilitate its function. On one side of the reader protein is a chromodomain that facilitates preferential binding of this reader to H3K9ME3 marks. And on the other side is a chromoshadow domain that facilitates interactions with other HP1 readers in spatial proximity. So as a result of interactions at the chromoshadow domain, HP1 readers have the ability to bridge nucleosomes in spatial proximity and pull these nucleosomes closer together. So right now we're looking at cryo-EM images of dinucleosomes, which are bridged by HP1 readers. These readers can pull these nucleosomes in a way that facilitate condensation of the chromatin fiber. And as a result of oligomerization of HP1 readers, we see that regions rich in H3K9ME3 marks, which are preferentially bound by HP1s, tend to compress into these puncta. So like I mentioned earlier, we develop physical models that study how patterns of epigenetic marks and interactions between their reader proteins dictate chromatin structure. We operate at a number of different length scales, including base pair resolution models and full chromosome coarse-grained simulations. And we're also interested in modeling how epigenetic mark domains persist over time. During this talk, I want to focus on the first 2 points, how chromatin organizes at snapshots in time. And I'll begin by focusing on the project that was inspired by last year's symposium on how clusters of nucleosomes form as a result of HP1 interactions. So as we heard earlier, the structure of the chromatin fiber is actually far more heterogeneous than we had previously thought. Rather than nucleosomes adopting this traditional 30 nanometer fiber arrangement, we see that nucleosomes they form sporadic clusters throughout a euchromatic phase. And these clusters are quite heterogeneous. They vary in size, and there's also heterogeneity in the amount of clusters that form in different regions of the chromatin, of the nucleus. So our goal is to develop a model that captures this clustering phenomenon, and we'll base this model on residual amounts of H3K9Me3 marks that are found in Euchromatin, as well as local HP1 interactions at these marks. I want to build this model step by step for our understanding. So we begin with a 1D array of nucleosomes. Each nucleosome has 2 histone tails, and these histone tails can be modified by H3K9Me3 marks. I'll go into how we establish these marks on the next slide, but for now, let's take that for granted, and we know that HP1 readers preferentially bind these H3K9Me3 marks, and these are the reader proteins that are responsible for affecting chromatin structure. So I mentioned that our nucleosomes are modified by H3K9Me3 marks, and this gives each nucleosome an identity. These identities indicate whether 0, 1, or 2 histone tails are modified with H3K9Me3, and we'll denote that with the variable S. So based on our analysis of ChIP-seq data, we've determined that the pattern of H3K9Me3 marks in euchromatic regions tend to be exponentially correlated. So where you see an H3K9Me3 mark at one site, you're more likely to see a mark at a neighboring site. So we sample from these exponential distributions to create synthetic patterns of epigenetic marks, and we fix these marks for the duration of our simulation. And using the sampling approach, we can create ensembles of epigenetic marks to see how the amount of the mark in the nuclear environment affects chromatin structure. So I mentioned, and I showed this cryo-EM image earlier about dinucleosomes being pulled together as a result of HP1 bridging. This is a distance-dependent phenomenon which is based on the linker length of DNA. So we need to find a way to model linker lengths along a 1D array of nucleosomes. So we have our linker lengths, Li, which indicate the amount of base pairs separating nucleosomes I and I plus 1. And we sample these linker lengths from an exponential distribution in the absence of HP1 interactions. And the average of this distribution is 45 base pairs. Because HP1 bridging requires nucleosomes to be in proximity, we set a limit on the range of these interactions to adjacent nucleosomes separated by less than or equal to 15 base pairs. Beyond 15 base pair linkers, the amount of energy incurred by bending the linker DNA to get these HP1s close together is greater than the thermodynamic benefit of the HP1 interactions. And based on the distribution of linker lengths and the energy difference between HP1 interacting configurations versus no HP1 interacting configurations, we can use Monte Carlo simulation as a way to sample our linker lengths along this chromatin fiber. Monte Carlo simulations require some sense of energy or favorability of chromatin states, which we denote with a free energy. In this free energy, we need to assign nucleosomes or linkers as facilitating HP1 interactions or being too long to facilitate HP1 interactions. So we denote this with our variable gamma. Gamma I is just an indicator variable, so it's equal to 1 if nucleosomes I and I plus 1 can form an interaction, and it's equal to 0 otherwise. And based on the free energy of configurations with and without interactions between nucleosomes I and I plus 1, we can sample linker lengths. So we know for random nucleosomes I and I plus 1, what's the energy difference between configurations with and without HP1 interactions. And we can bias our linker length distribution to account for this energy difference so that we have a preference for shorter linker lengths which facilitate HP1 bridging. And we can iteratively sample linker lengths using this biased probability distribution until we converge on a profile of linker lengths for a 1D array of nucleosomes. And in this, so this is showing a profile where on the y-axis we have our linker lengths, and on the x-axis we have a position along a 1D chromatin fiber. And we see that mark domains tend to have shorter linker lengths because these marks are preferentially bound by HP1s. These HP1s want to oligomerize with one another, so they tend to move the nucleosomes closer together. So far, we've been operating in a 1D space, but really we're interested in the structure of chromatin in 3 dimensions. And for this, we turn to polymer theory. So we assume that the linkers between nucleosomes behave as semiflexible polymers. This is based on the behavior of bare DNA. And we can sample bending probabilities of these polymers so that we grow chains of 3D chromatin configurations based on the linker lengths between nucleosomes. So we have this chain growth algorithm that's very efficient, and we can generate thousands of realizations for a single linker length profile. And from these realizations, we can evaluate cluster size distributions. How large are these nucleosome clusters that results from residual amounts of H3K9 Me3 marks in euchromatin. So in this model, we have 2 parameters that we can play with. We have the amount of our H3K9 Me3 mark, and we have the amount of our HP1 reader protein. And as we increase the amount of H3K9 Me3 marks, we see that there are more favorable sites for HP1 to bind, and we get overall increased compaction of the chromatin fiber. Likewise, if we increase the amount of HP1 in the nuclear environment, there's a greater pressure for these HP1s to bind the chromatin fiber, and this also leads to a more compact configuration. So we can vary these 2 parameters simultaneously to evaluate how changes in our H3K9 Me3 concentration and in our HP1 concentration affect chromatin structure. And we see that as we increase the amount of H3K9 Me3, or we increase the amount of HP1, we get larger, more compact clusters. So this is a fine model if we can demonstrate that it's experimentally valid. And to do this, we look to work from Claude O'Shea using Chrome EMT to generate in vivo images at nucleosome scale resolution. So they've generated tomography slices of the euchromatic domains and evaluated cluster size distributions from those slices. And we're interested in comparing our models to results from Chrome EMT based on tuning biologically relevant parameters. So we know in the nuclear environment we have a shared pool of HP1s floating around available to bind the chromatin fiber, but we also know that there's spatial heterogeneity in H3K9 Me3 marks. So to validate our model, we want to fix the HP1 concentration, and we want to vary the amount of H3K9 Me3, and we want to see if we can replicate the cluster size distributions generated using Chrome EMT. And we show that we are able to do just that. So on in the histograms in cyan, we see the results, the cluster size distributions that were measured from images generated by Chrome EMT. And on the right, we show the cluster size distributions generated by our model. So in summary, we have a model that has 3 key ingredients. We have correlated patterns of H3K9 Me3 marks. We have preferential binding of HP1 to those marks. And then we have oligomerization or favorable interactions between HP1s in spatial proximity. We know that varying these parameters affects the clustering along the chromatin fiber. And by varying biologically relevant parameters, we were able to replicate conditions measured experimentally. So the take-home message is that, according to our model, heterogeneity in H3K9 Me3 marks cause heterogeneity in HP1 binding, which results in this cluster formation. It looks like we still have some time for the second part of the talk. So I want to go into our more recent work studying full chromosome compaction. So how do larger domains of nucleosomes tend to compartmentalize as a result of multiple epigenetic marks and epigenetic reader proteins. So for this talk, I want to introduce a second reader protein, PRC1, which, like HP1, has a preference for a specific epigenetic mark. This epigenetic mark is H3K27ME3. So the PRC1 readers have a chromodomain similar to HP1, which preferentially binds H3K27ME3 marks. And then PRC1 also has a mechanism for interacting with other PRC1s in spatial proximity. So these are head-to-tail interactions at the PHC subunit. And as a result of these interactions, we see puncta form around transcriptionally suppressed regions of chromatin, which tend to be rich in H3K27ME3 marks. So there seems to be some sort of cross-interaction between these two epigenetic marks. Back in 2003, a paper showed that HP1 binding increases in the presence of H3K27ME3 marks, and similarly, PRC1 binding increases in the presence of H3K9ME3 marks. So this is a mismatch between the reader protein and the epigenetic mark, but the presence of this mismatched reader protein, the presence of this mismatched mark makes it easier for the reader protein to bind. Similarly, in 2014, a model was proposed by which the writer of H3K27ME3 marks facilitates binding of HP1, and they demonstrate that up to a 6-fold increase in HP1 binding happens in the presence of H3K9 and H3K27ME3 marks compared to H3K9ME3 alone. So we're interested in developing a model that captures this cross-interaction, or that gives us a way to experiment with how cross-interaction affects chromatin structure. And again, we begin with an array of nucleosomes with 2 histone tails on each. And this time we modify these histone tails with H3K9 and H3K27ME3 marks. We do so based on ChIP-seq data instead of sampling from a distribution this time, since we have coarse grain data equivalent to that of our model. And then we have our 2 reader proteins, HP1 and PRC1, which preferentially bind their respective marks. The way we capture this binding is by evaluating a binding free energy, which we denote with F. So sigma in this equation gives us the amount of bound reader proteins on nucleosome I. This binding is influenced by the degeneracy of a binding state. So we have states 0, 1, or 2, which don't tell us exactly which histone tail is modified by a mark. So we need to account for indistinguishable states with this degeneracy term. We also have a bias for binding, which is given by a chemical potential, which tells us if we have more reader proteins in the nuclear environment, there's a pressure for those reader proteins to bind the chromatin fiber. And we have to take that into account when evaluating the probability of a binding state. And then lastly, we have the preference for a reader protein to bind its respective mark. So we can evaluate for any configuration a total binding energy given by the sum of the binding energies for HP1 readers and PRC1 readers at each nucleosome I. And then once these reader proteins are bound, in order to affect chromatin structure, they need to interact with one another. So we capture this interaction between reader proteins using a field theoretic approach. So traditionally, if we're interested in modeling long-range interactions, one approach could be to evaluate pairwise distances between nucleosomes and then add an interaction energy for any pair of nucleosomes that are within a certain distance. But for full chromosome models, the complexity of that type of calculation is intractable because pairwise interactions scale as an order of n squared, where n is the number of nucleosomes. If we take this field theoretic approach, we can reduce that scaling down to order of n, which is tractable. And the way this works is we have our 3D positions of each nucleosome on the chromatin fiber, and we want to distribute these nucleosomes as weights into voxels, discrete voxels that make up our space. So we use trilinear interpolation to distribute the weight of each nucleosome into nearby voxels. In this diagram, I'm showing a 2D example of that just for interpretability. And then the energy of interaction scales with the density within each voxel. So where we have larger concentrations of our reader protein, we tend to have more interactions. and that leads to a larger contribution to the interaction energy. So the interactions originate from HP1-HP1 bridging. We have our PRC1 oligomerization, and then we're introducing this HP1-PRC1 cross interaction, which give us a variable we can tune over the course of the simulation. Going into finer details, if we have two reader proteins bound to the same nucleosome, we know that they will interact because they have to be in spatial proximity. So we remove those reader proteins or their contributions from the field, and we add a direct interaction term. So far, we've introduced a binding energy and an interaction energy. And the last ingredient in our formula is the bending probabilities of the linker DNA between adjacent nucleosomes. So I mentioned this in the first half of the talk. We assume that linker DNA, is a semiflexible polymer, which means it resists bending at short length scales. So there's contributions to the favorability of a state based on the bending of that segment. We have the stretching of that segment, and then a shearing effect of that segment, with a correction for how bending and shearing kind of capture the same information. So now we have a free energy equation that captures contributions dictating the favorability or the likelihood of a chromatin state. And we can sample chromatin configurations using Monte Carlo simulation based on those free energies. So we apply random geometric transformations to segments of the chromatin fiber, and we evaluate the energy change associated with each transformation. What Monte Carlo simulations mean are that favorable moves that reduce the free energy of the system or make a chromatin fiber more likely, are always accepted, while moves that increase the free energy are probabilistically accepted. So with Monte Carlo simulation, we can vary the parameters in our model to see how the structure of the chromatin fiber responds. The parameters we're interested in are the concentrations of our reader proteins, both HP1 and PRC1, as well as the self-interaction of our PRC1 readers, and the cross-interaction between HP1 and PRC1. We know that HP1-HP1 interactions tend to contribute a constant energy just based on previous work from our group, so we use that constant value to reduce the dimension of our search space. But we still have a 4-dimensional search space, and these are all continuous variables, which become intractable to search using a grid search method. So the way we approach this is by picking random values for these parameters, and then training a multi-layer perceptron to interpolate an outcome of interest between the simulated values. And one demonstration I want to go into, this will be the last part of this talk, is focused on predicting HP1 binding. So if we assume that there's no direct crosstalk between HP1 and PRC1, one null hypothesis could be that HP1 binding is independent of PRC1. But if we vary the amount of the favorability of PRC1 interactions, or the concentration of PRC1 readers, we see based on our model that there's an implicit crosstalk that happens. So the conditions governing PRC1 interactions affect the binding of a different independent reader protein. And the reason for this is that both of these reader proteins bind the same chromatin fiber and affect the same environment. So if one reader protein condenses the chromatin fiber, HP1s can find other reader proteins, other HP1s, more easily, and this makes it more favorable to bind. So in conclusion, we've developed a model that captures the interaction between multiple epigenetic marks, and we've used this model to predict some behavior in the governing chromatin organization, specifically how different reader proteins seeming to be independent actually interact through an implicit mechanism. And with that, I want to thank our group. I want to thank the Center for Physical Genomics and Engineering for the invitation again, as well as our funding sources.